Western blot is a fundamental biochemistry technique we can use to detect the presence and relative amounts of proteins that we're interested in. So basically how it works is we take a sample that contains a bunch of proteins, maybe you break open a cell and take that lysate, and maybe you do it from cells under different conditions, like with or without a drug, or different cell types that you want to compare the expression of this protein in. Then what you're going to do is you separate all of the proteins in that sample using an SDS-PAGE gel. So basically you denature or unfold the protein, coat it in this negatively charged SDS detergent, send it traveling through this gel, and then the proteins are going to separate by size. And so you have a bunch of proteins like separated by size through this gel because the longer ones are going to travel slower because they get stuck more in the gel. So it's going to take them longer, and you turn it off. And if you were just to like stain the gel now with like Coomassie, so with like a general protein stain, you would see a bunch of bands, and each of those bands would be corresponding to various protein or proteins that have a similar size because you're separating them based on their size of their denatured chains. But that doesn't tell you anything about the abundance of a protein of your interest. That just tells you about like a general size distribution of various proteins in your sample. And so we use SDS-PAGE a lot, especially when we're purifying proteins, because you want to see that you're like getting rid of excess bands. But right now, what we're trying to see, like, is this specific protein? And we don't have any information about the identity of the proteins at this point. Just if we were to stain it, we can see the relative sizes of the proteins in the mixture. And there's a bunch of proteins that look about the same size, so we have no idea what's going on in this sample. So what we need to do is we can actually test for the presence of specific proteins using antibodies, so little proteins that are going to recognize and bind to the protein of interest. But right now we just have all these proteins just like hanging out in this gel. So we turn off the electricity so they don't have that like charge gradient helping push them through, pull them through, or whatever, motivate them to go swim through this gel. But they can still just like kind of like diffuse out, so like move around and stuff. And plus the gel's like flexible. It's like a little thin little thing, and trust me, those rip a lot. And so what we need to do is we need to transfer this to something more stable in order to bind the antibody to it. And so what we do is we're going to transfer it to a membrane, and we can do this with the Western. This is the way the Western blot set comes in. So we make this little like membrane sandwich where we have some filter papers and some sponges to keep things all snug, and then the membrane. And we're going to basically transfer the proteins out of the gel onto the membrane. So the SDS-PAGE, we're running it this way. With this one, we're going to run it like through or this way. So we go down and then out onto this membrane. And then we can use these antibodies to test for the presence. We go probing, kind of like fishing for specific proteins of interest using these antibodies. And so a drawback is that you can only detect things if you're looking for them. You're going fishing, but you're using bait that only one type of fish likes. So you get information about that, but not any idea of what else is going on. So please, 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 if you're doing a Western blot, also show like the SDS-PAGE, like the Coomassie version. So take some sample, do a Coomassie, some with your SDS-PAGE or with your Western, especially if you are doing like a purification. You're trying to say like, oh, I purified this protein, and then you show me a Western blot. No. Okay, so let's go into how this all works, and then some more specifics. Just a little note that this is a recut version of a post I made last year, so I added a little bit of new stuff, and directing you to some other of my posts as well, where I have more information about the various topics and techniques, also how this fits into what we've been talking about measuring protein expression. So I'm going to give you an overview, and then we're going to go into more detail in each of the steps. So basically the idea is that you take proteins from different conditions. So maybe you have cells before you add a drug and cells after you add a drug. Then you're going to start off like we did with the SDS-PAGE. You're going to, so you need to break open those cells, isolate the proteins. Then these proteins are all going to have different charges and sizes. And so basically you're just going to use the detergent SDS to unfold all the proteins, keep them soluble, and give them a negative charge that's going to allow the electricity to get them to travel through the gel mesh. So that's the gel electrophoresis step. But then you have your proteins locked in this gel, which isn't very useful for Western blotting, because you're going to have to be doing a lot of binding and washing and binding and washing, and these gels are really thin and they're just going to rip. So what you need to do is you need to transfer them to a more sturdy membrane.
So this is the blotting step, or the transfer step, and there are different ways to do it. I'm going to tell you about the way that I've done, which is wet electrotransfer. So at this point, you have your proteins in the gel, and you need to be able to see if there are specific proteins in there. But so for reasons I will talk about, you first need to block the membrane. So basically, right now, your proteins are on this membrane that is basically kind of like duct tape for protein. So it's going to stick to basically any protein. And so we want it to stick to the proteins that we've just transferred, but they're already there. But the protein, we're going to use antibodies, which are proteins, as probes to see if our protein of interest is there. So the antibodies, if there are free spots on that sticky tape membrane, they're just going to stick, and then you're going to get high background. So we don't want them to stick everywhere. We only want them to stick where our proteins are present. So we block the membrane using a generic protein and some detergent to kind of make things more slippery, so that the antibodies can't just generically bind. And then we're going to bind with a primary antibody that's going to recognize directly the thing you're interested in, and then a secondary antibody that's going to bind to that primary antibody. But it's also, the reason for this is that this way you amplify your signal, because you can have a bunch of secondary antibody binding to the primary antibody. And the secondary antibody is attached, or what we call conjugated, to something that's going to make it able to visualize it, such as some fluorescent protein. And then you're going to wash. Yeah, there are a lot of washes because you want to make sure that you're only getting the specific things, washing away anything non-specific, because you flood the membrane with antibodies, and so you want to get rid of those antibodies before you go to screen it, and then you visualize it to see if your protein is there and the relative amounts. So that's the overview we're going to be returning to throughout, and I'm going to show you the steps in more detail. So starting with this beginning part, so we're going to take our cells, and you don't have to start with cells. If you can start with like some purified protein mixture or that sort of thing, but often you're starting with cells, so you want to lyse them, which is where you break them open. And so now you get this mix, and you get rid of the membrane parts by like centrifuging. So you spin them really fast so that the membrane parts pellet out, and now you're left with this mix of soluble proteins. But they're all folded up, and they all, if you try to send them traveling through a gel, because they all have these different shapes, they're not going to uniformly travel based on their length. So it'd be hard to compare their sizes. Also, the way that we get the proteins to move through the gel, so the electrophoresis part, it relies on the proteins being negatively charged, because basically you use a charge gradient where you have a negative charge at top that's going to repel the negatively charged proteins, and a positive charge at the bottom that's going to attract the proteins. So we want all the proteins to be negatively charged and unfolded. So the detergent SDS is really great for this because it has this long, like greasy, nonpolar tail, and this hydrophilic, water-loving, negatively charged head. So when the protein unfolds, it kind of like globs onto the protein and keeps it soluble in its unfolded state, and also gives it this negative charge. So this is going to allow us to separate the proteins by length in this gel electrophoresis step. So basically, in this first step in SDS-PAGE, we're sending the proteins traveling vertically through this gel. And you can see in this picture that they're separating by size. So basically what happens is the proteins are kind of going to get tangled up in this gel mesh. And the longer they are, the more tangled they're going to get. And so the slower they're going to travel. And so when you turn off the electricity and prevent them from moving through the gel, the bigger ones are going to be higher up. So that's what's happening. But what you're seeing is basically nothing. You're just seeing the tracking die. When you're doing a Western blot, you probably have a stained ladder though, that you'll be able to see, and that stained ladder is going to be important for making sure that your protein's transferred to the membrane. So you start the same way, whether you're just doing like a normal SDS-PAGE or you're doing a Western blot. So with a quote unquote normal SDS-PAGE, what you might do is Coomassie staining, which is an all protein stain. And so you're going to see bands for all the proteins that are present. But you don't know what those bands are. So you can get a general idea of their size, especially if you use a protein ladder, which is basically a mix of proteins of known size. So you can see that on the left hand lane. So that allows you to compare the protein sizes, but it doesn't tell you what they actually are. So for that, you can look for Western blotting, which is what we're going to talk about now. And that's going to show you specific proteins. It's often with like some fluorescent readout or whatever. So you might get something that looks like this, and you can see the thicker bands correspond to more protein, but it's not like very quantitative or anything. Also, an important thing that I'm not showing here is that you're going to want to like also stain or also probe for a protein that should be the same in all the samples, like some housekeeping protein like GAPDH or tubulin or something. That's like a loading control, so that if you have a stronger band, it's not just because you loaded more. If it was just because you loaded more of that sample, then you would have more of that loading control as well. But if you have the same amount of loading control but more of your protein of interest, that means that the protein is like being expressed more or degraded less or something in those cells. So first we need to transfer it. So we're going to blot the proteins. We're going to move them to a membrane. So in SDS-PAGE, we had the proteins go down, and now we're going to have the proteins come out. And so this is showing a wet electrotransfer setup. And so basically, these use the same electrophoresis module, like the bucket outer part, but a different like insert module part. But there are different ways of electrotransfer. There's like dry and semi-dry and stuff. So and some of those are like horizontal and stuff. So I'm not going to talk about those. So this is the way that we do it. And basically what you're going to do is you have this sandwich you make this like sandwich with gel and membrane and sponges and filter paper, and you're going to stick it in that module, and it'll, so this way, it's confusing because it looks just like kind of like the proteins would be going down, but the way that the electrodes are set up, the charge gradient is actually going like horizontally. So you're taking things out of the gel onto the membrane. But in order to do that, you need to make sure that you set up your sandwich right. So basically, you have this like cassette, and you want to make sure, so you have filter papers to help keep things wet and wick the buffer through, because the important thing is that you need to have the charge be able to flow consistently. Like you need to have you don't want to have any bubbles or anything, or any dryness where the electricity can't travel through, or you're going to get weird artifacts on your gel and things aren't going to transfer. You need to have like a conductive path for these proteins in the charge to move. So that's why you have the filter paper, and why it's important that when you're making your sandwich, you want to make sure that everything stays wet the whole time, so you like make it underwater and stuff. So after your filter paper, you're going to have your gel, which has your proteins for now, and then you have the membrane you want to transfer them to, some more filter paper, some sponges to keep things snug. You don't want things shifting around, or else the proteins aren't going to be where you expect them, and you don't want things just like diffusing out. But the most important thing is that you put your cassette into the thing in the right direction. So you want to make sure that you have the membrane in front of the gel, in the way that the electricity moves in your module. So once you set it up, you're going to wrap it all up and slide it in. Fill up the tank with your transfer buffer, hook up the power. Then you're going to get something where when you open this sandwich thingy up, your gel should be empty, and the protein should be on the membrane. You're not going to see the proteins, and you're not going to see them in the gel, and you're not going to see them on the membrane. But what you can see is the pre-stained ladder. So normally when we're just running like a normal SDS-PAGE, we'll use a cheaper ladder, which isn't pre-stained. And you just stain it the same as you do your proteins, and so you're just relying on the loading buffer to track the run. But here it's important that you see that the proteins actually get transferred. So if you use a pre-stained ladder, then that pre-stained ladder is going to get transferred from the gel to the membrane along with all the other proteins. So you shouldn't see the pre-stained ladder should all be on that membrane and not on your gel. The amount of time that you want to use to transfer is going to depend on like how big your protein is. The bigger it is, the longer it's going to take to travel, and you might need to like up the voltage or up the time or something. But if you do that too much for a smaller protein, might actually like come out of the membrane, so you have to kind of optimize for your protein of interest. Then to find that protein of interest, you're going to use antibodies. So now we're at this stage. So an antibody is basically like a little protein that recognizes a specific part of something else. And so we call that something else an antigen, and we call the part that gets bound by that antibody an epitope. And so we say like recognize, but really that just means that they bind specifically to it, or you hope it's specific. Sometimes they can be non-specific, and then you can get noisy signals. So I like to think of it kind of like Where's Waldo. So each of these antibodies has like a constant region, which is in this case they're different. So they're different types of antibodies, but and we're going to talk about these IgG antibodies where they have this like Y shape. And so the base of the Y is like a generic part that's specific for that species. And then the tips of the Ys are the variable parts, and that's where the antigen is going to bind. So those variable parts can recognize different parts of a protein, like recognizing different parts of Waldo. So if you have what's called a polyclonal antibody mix, that's going to recognize, you're going to have a mix of antibodies that can recognize different parts of Waldo. If you have monoclonal antibodies, that means you just have one of these antibodies. So this can have better specificity, but lower sensitivity. We'll get to that more later, but basically, if you recognize more parts of Waldo, you're more likely to also recognize similar parts in other proteins, and thus get lower specificity. So you're recognizing other things, and you're getting false alarm signals. But if you have a bunch of different antibodies that recognize different parts, then there are more places the antibody can bind on your protein, and thus you can have a stronger signal. And polyclonal antibodies are also often cheaper. So if you were looking before, we saw there was this primary antibody and secondary antibody. So your primary antibody is going to have a generic base that's generic for the animal that made the primary antibody. So this could be goat what's a protein that you're wanting to look for? Goat, we're just going to make up a protein, anti-fabulous. And so we're going to have this protein fabulous. And so our primary antibody is going to be goat anti-fabulous. So the base part is going to be generic for goat. And then at the top, we're going to have that specific part that specifically binds to fabulous protein. So you get something like this. So it's useful because it bound to our fabulous protein, but it's not useful because there's only one per protein and because we can't see it. So the whole point of the probing is to make it so that we can see it. So for this, we need a secondary antibody. So this is going to be made in a different animal. So maybe this is like rabbit. So this would be rabbit anti-goat. So it's going to recognize the generic part of our first antibody. And this is good because we can use it with goat antibodies for any protein. So we can save money because we can use the same secondary antibody for any antibody that was made in a goat. We just can't use a goat secondary antibody. You have to use a different species. So we have this secondary antibody, which is going to recognize the generic part of our first antibodies. and it's going to be attached, or what we call conjugated, to something detectable. So this could be like a fluorophore, so something that's going to give off light when you shine a light of a different wavelength of it, or it can be some like chemical reactant group that if you add chemicals, it's going to undergo some sort of reaction that's going to make it a colored product. So an example of this is like horseradish peroxidase. So the secondary antibody is going to bind to your primary antibody, and then when you add those chemicals or you shine that light, you're going to get some sort of signal that you can see, so often fluorescence or some sort of color change. So, okay, so that's the idea. But we skipped a step in our explanation, because if you were just to do that, you would get really high background. So you'd have a lot of crying wolf, basically. So as we talked about in the very beginning, our membrane is kind of like sticky glue for protein, or like duct tape for protein. And so our antibodies are proteins. So our antibodies are just going to stick to that membrane unless we block it. And so basically blocking just means we take like a generic protein like BSA, bovine serum albumin, or even milk. A lot of times you make a milk solution with powdered milk, and often detergent. So the buffer is often like TBST, so tris buffered saline with tween. I think it's tween, not trine. But basically that's a detergent. So you can think of it kind of like when your hands are slippery, it's harder to grab things, so it's harder to stick to things. Actually, my cousins and my sister and I, we got locked in a bathroom once because we weren't very smart. We were little at the time, but our hands were all wet and soapy, and we couldn't open the door because our hands were so slippery. So it's kind of like that. Like if the membrane's really slippery, the antibodies aren't just going to bind randomly, or that's the hope at least. And then you also have all those wash steps to see, so that if they do bind, then they'll get washed away. And so the wash steps are like the worst part of all of this, because it's just really time consuming and boring because you keep having to wash and wash and wash. But it's important to wash, or else you'll get high background. So yeah, so that's the basic idea of that. Some other things to look out for. So cross-reactivity. So this is what we're talking about with the monoclonal versus polyclonal antibodies. So if you have polyclonal antibodies, there's more of a chance. So that's where you have antibodies that recognize like different parts of Waldo. So it's more likely that you're going to recognize other things that also have those parts, or something similar. So you're going to have higher cross-reactivity. But then you also have the sensitivity, where if you have a polyclonal, there are more places to bind, so you can get a stronger response and signal. But with monoclonal antibodies, there's only one place to find, so you're going to have lower sensitivity. Although with the secondary antibodies, you'll get somewhat of a boost with either method. A little troubleshooting. So yeah, so you want your antibody to only bind the protein of interest. If your antibody doesn't bind, then doesn't bind to your protein. This can actually happen. Not all antibodies are made alike. Some antibodies are a lot better than other antibodies. You want to make sure that if you have like a positive control, so a protein that you know is that protein that it should be able to recognize, that's a good, you want to include that. So like if we had to sample a pure fabulous protein, we'd want to include that to make sure that the antibody could recognize it. So antibodies recognize proteins based on their shape, kind of. So in a Western blot, our protein is unfolded. So normally what you want is you need to make sure that the antibody that you're using is can recognize the unfolded form, which usually it can. But like sometimes people use antibodies like in cells, so where the protein's folded, but you need it to be able to recognize the unfolded form. Antibodies can also expire, so you want to make sure that you store them properly, because they're they're proteins, so they can degrade, and that sort of thing. So you often store them at -20 or -80. You can also store them like diluted for shorter periods in the fridge, because you can actually reuse the solution a couple times. So if you're getting high background, you want to make sure that you're adequately blocking your membrane. If you're getting kind of grainy signal, you want to make sure that if you're using powdered milk, you want to make sure that milk is really dissolved, or else you'll get grainy artifacts. And don't use milk when you're detecting phosphoproteins or a biotin-based detection system. So basically, if your milk has a bunch of like phosphorylated proteins in it, and so it's going to cause problems if you're trying to stain for phosphorylated proteins.
One other word of caution is that you can also get problems with your Western blotting if you have bubbles. And so you don't want to have like air bubbles in between like your membrane or your gel, because then that's going to impede the transfer. So you also don't want to go overboard when you're trying to avoid bubbles. So I remember in undergrad when I was learning how to do a Western blot in my biochemistry class, thank you, Dr. Vidya. She was teaching us how to avoid bubbles. And so you do everything when you're preparing your sandwich, you're doing it all in like a dish of like the transfer buffer. So you're not letting any air in, and then you roll out. As you're doing this, you're rolling with a pipette to remove any air bubbles. And so worked great in undergrad, and I took that spirit with me to grad school. And then in my first grad school rotation, I was doing a Western blot, and I was rolling out those bubbles, and it turned out that I was a little too overzealous in my rolling, and I ended up with this very interesting looking gel or looking Western blot. So the moral of the story is you want to roll out the bubbles, but do it carefully and gently, so that you don't like end up tearing your gel or like tweaking the angle of your gel, or you're gonna get artifacts. So of course, this was right before I was supposed to like present a lab meeting, and I didn't have time to like redo this or anything. And so I got to present this beautiful quirky gel, and to make me feel better afterwards, the PI, so the head of the lab I was rotating in, he told me that he actually had a paper where he had to piece together the pieces of a gel because it had fallen apart, and it was like this huge gel that was a ton of work. So we had to actually like piece together the pieces of this gel, and it still made it into this like groundbreaking paper. So I felt better after that, less embarrassed, and know that this sort of thing happens to everybody, but you can learn from your mistakes. And so if you want to learn from more of my mistakes, I have a post with tips about that. So to put things in context, Western blot is just one of the many techniques that we can use to measure quote unquote expression. So when we talk about expression, you can mean different things, because to go from the genetic recipe from the original gene to the protein, there's a lot of steps and there's a lot of places and opportunities for regulation. So you can look at these various intermediate products as well as the final product in order to get ideas about how the gene is being expressed. And with a Western blot, we're seeing basically the contributions of the protein being made and the relative like protein being made, as well as you have to account for the fact that the protein can get degraded. So even if you're making a lot of it, you might be degrading some of it. So yesterday we looked at another technique. We looked at RT-qPCR, which is measuring messenger RNA levels by making a lot of copies of them. And then measuring those copies as they're made. And the more copies you start with, the faster the fluorescence is going to rise. And so that's one technique that we can use where we're looking at the messenger RNA level. So that's like a step before the protein level. But there can also be this is going to depend on transcription as well as mRNA decay. And then when we're looking at the Western blot, we're measuring protein levels, which is going to, as we said, reflect the contributions of all of this, as well as protein degradation. And so a lot of times what people really care about when they're talking about expression is that final protein product. And Western blot is a way to test for this. It's also not the only kind of blot. And so if we go back to measuring messenger RNA levels, there's this thing called the northern blot. And the northern blot, it's going to look for specific messenger RNAs. And it's going to do this in a kind of similar way to the Western blot, except here we're separating RNAs by size. Then we're blotting them onto a different type of membrane. And then we are going to probe using a radioactive probe DNA. So we have a labeled DNA that matches that RNA. It like complements it, and it's radioactively labeled or fluorescently labeled. And then we get it to stick, and then we see how much of that RNA is there. So this is just, there's a lot of other blotting techniques, and I should mention that Western blotting, technically it should be a lowercase w. The Western isn't for its name, it's for basically it's a pun. The first blot is the southern blot, which is a DNA blot that does kind of like what we were talking about for the northern blot, except we're using DNA to probe for DNA instead of DNA to probe for RNA. So this guy was actually named Southern. And so the technique with the southern blot, you get this capital S. And then to kind of be punny, people started naming all of these other types of blots after compass directions. And so those ones shouldn't be capitalized, although people capitalize them very commonly. And I know I do sometimes. And people won't get mad at you, I don't think. No one's ever gotten mad at me for miscapitalizing it, although I try to make a point to decapitalize it. But as you saw in that other post, I accidentally capitalized northern blot. So yeah, but nobody's perfect, right? Speaking of tips and stuff, I have a lot more on my blog. And so I have a ton of text and static figures for what we talked about today in my Western blot post. I also have a more practical Western blot video if you want to see really more hands-on what it looks like. And I have a whole page of posts, like links to posts I've done on various techniques. So a bunch of things we're talking about today, as well as some other stuff. You can find it under Let's Talk Science Lab Techniques, and I've got a bunch of other stuff in here too. And I'm not trying to I get nothing out of this other than being able to help people. So I'm not trying to get money. I'm not trying to do anything. I always feel weird directing people to my own stuff, but I'm just doing this to help people. And so I want as many people as possible to be able to find these resources. So voila, they're there if anybody is interested. And so yeah, so hope this helps people, and thanks for sticking along with this kind of recut edition. So I didn't have time to redo the whole thing, and I liked what I'd done before, but I just thought I'd add a little more. So hopefully that was okay with everybody, and if not, I'm sorry. So yeah,